I have posted a video on the movement of information from DNA to protein, which is the final form. Please make sure you go back and watch that video before you proceed with the next slide. So this slide over here shows how protein synthesis occurs in the cell. This ribosomal structure is the bead-like structure that you saw floating around all over the cell. And ribosomes are the site of protein synthesis. So ribosomes are present on the surface of the endoplasmic reticulum. So all over the endoplasmic reticulum, you can see little bead-like structure. Little bead-like structures known as ribosomes. So the structure over here is the endoplasmic reticulum. Ribosome makes the protein while it is attached to the surface of the endoplasmic reticulum, and then the synthesized protein is folded into the desired shape. So this is called protein modification. And in the tunnel of the endoplasmic reticulum, the protein is folded and they might have some modifications attached to it. And the protein is packaged into tiny membranous structure known as vesicles. So these vesicles pinch off from the endoplasmic reticulum and are transported to the Golgi apparatus. The Golgi apparatus is like the USPS of the cell. So the Golgi apparatus receives the protein from the vesicle, and then it further modifies the proteins by adding some protein molecules or amino acids or sugars to the surface. So you can consider those as the shipping labels of the protein. Sometimes the protein needs to be shipped into the cell towards the nucleus or towards the plasma membrane that usually goes out of the cell. Now, proteins on the surface of plasma membrane are usually directed by the Golgi apparatus to go towards the plasma membrane. And if there's a protein that is necessary for function inside the cell, the Golgi apparatus will put tiny zip codes of amino acids or sugars on the surface of the protein in order to ship it into the cell. Alright, so the next type of endoplasmic reticulum is the smooth endoplasmic reticulum. They lack ribosomes and they usually help in storage of lipids and breaking down of lipids. And like I said, they also have some toxins of drugs and pesticides that are stored in the smooth endoplasmic reticulum. So if you look at the liver of somebody who's really alcoholic, you can see that their liver cells would have evolved to have several more smooth endoplasmic reticulum than a person who does not drink. Similarly, a person who takes a lot of pills will have a lot of smooth endoplasmic reticulum for detoxification of drugs and pesticides. So keep in mind, structure defines the function, and different types of tissues or organelles may have more or less of a particular organelle depending on their function. So like I said, Golgi apparatus are the shipping centers of the cell. So if you take a look at the Golgi apparatus right over here, Golgi apparatus are a part of the membranous structures that emerge from the cell. So right by the endoplasmic reticulum, there is the nucleus, and along the nucleus you have the rough endoplasmic reticulum, which are studded with ribosomes. After the endoplasmic reticulum in the cytosol, you have specialized structures known as the Golgi apparatus. The Golgi apparatus are stacks of flattened membranes that have associated vesicles with it. So they are either capable of receiving vesicles from the endoplasmic reticulum, and once they receive the vesicles, they modify it or put some zip codes in the form of amino acids or sugar molecules And then again, they package them into membranous vesicles and ship them either towards the plasma membrane to embed the cells with, protein molecules, cell membrane with protein molecules. If you remember, the plasma membrane contains a bunch of proteins. So these secretory vesicles that contain protein molecules may either end up in the plasma membrane, or some of these protein molecules may end up inside other membranous structures, other organelles of the cells like lysosomes. We'll look at the function of the lysosome in just a second. And then the other possibility is that the Golgi apparatus may pass over the protein molecules towards a plasma membrane to be secreted out of the cell. So if anything that is being secreted out of the cell, the process is known as exocytosis, means exiting a cell. Now these are the three pathways. Keep in mind, the first one is a pathway that carries these vesicles that contain protein to be secreted out of the cell by exocytosis, or the vesicles can carry membrane proteins to fuse with the plasma membrane, or if these proteins are required by any organelle inside the cell, they can carry the proteins into the cell as well. So the endoplasmic reticulum manufactures protein, the Golgi apparatus modifies protein and shifts the protein to the right place. So, like I said, some of the proteins that are formed go into the cell. And one such example is the proteins in a structure known as lysosomes. Lysosomes are the garbage bags of the cell. So like us, like we excrete everything that is unnecessary and sometimes we sweat it out, lysosomes basically take everything that is disposed inside the cell and they break it down into smaller substances. So enzymes that are required by lysosomes are known as lysozymes. They contain digestive enzymes that degrade material that are no longer useful inside the cell. And if there's an organelle, just say this membranous vesicle is old and it needs to be broken down, it will fuse in with the lysosome, and the lysosome will break it down into smaller particles or substances. So once these molecules are digested, the liposomes, maybe they fuse with the plasma membrane and they spit out all the digested substances, or if they're like amino acids which can be reused by the cell, they may go back and be released into the cell to be reused by the cell. Alright, so the other structure that require proteins in the cell are peroxisomes. So peroxisomes are also membranous sacs that function in detoxifying in a cell. So apart from smooth endoplasmic reticulum, peroxisomes also function in detoxification inside the cell. So membranous sacs of peroxisomes contain specialized enzymes known as oxidase enzymes. Now our cells produce a lot of harmful chemicals known as free radicals. Free radicals are highly reactive chemicals that need to be neutralized, because if these free radicals are not neutralized, they end up destroying the cell. And free radicals are further converted to hydrogen peroxide by oxidase enzymes, and this peroxide is then converted to water. So this peroxide also is not harmless, because if you have ever seen or poured peroxide on a wound, it bubbles, right? It reacts with the wound and it destroys the cell. So peroxide and other free radicals need to be converted to water, which is not a harmful form. So the next part of the cell is the skeleton of the cell. So the skeleton structure of the cell is known as the cytoskeleton. Cyto means cell and skeleton is the skeleton which provides the structure to the cell. So the skeleton of the cell can be made up of 3 different types of filaments. And these 3 filaments together provide internal framework that determine the cell shape and support cell organelles. They help in intracellular transport. They also help in cell-to-cell attachment, or they can help in providing machinery for movement of cell organelles when necessary. So the major filamentous molecules of the cytoskeleton include microfilaments, intermediate filaments, and microtubules. So the microfilaments are fine, really thin filaments of protein, and they help in muscle contraction and several other types of cell movement if they are present. So since they are actin filaments, obviously the muscle cells will contain a lot of microfilaments. Okay, and then you have intermediate filaments. Intermediate filaments are protein fibers, and their composition varies from cell to cell. So they help in resisting mechanical forces that are acting on the cell and prevents the cell from being, folded too much. So they stabilize the structure of the cell. And the third type of filament are the microtubules. The microtubules help in giving the cells a shape, and they're involved in cellular movements and cell division. We'll see that microtubules basically help in forming something called as centrioles. And these centrioles help in the formation of thread-like structures known as spindle fibers that you will come across during cell division part of the lecture. So centrioles are a type of, they are made up of microtubules, and they are cylindrical structures which are made up of specialized proteins known as tubulin. So tubulin proteins make up centriole, and they help in formation of mitotic spindle during cell division. So these are the thread-like structure I was talking about. So keep the word centrioles in mind because you will come across this word when we talk about cell division again. Some cells also have extension structures outside the cells, and they can be majorly of 3 types. The first one is microvilli. Microvilli are really tiny hair-like structures that increase the surface area. So this is an example of a cell where you can see microvilli. Microvilli are present quite a bit in the small intestine because the small intestine functions in absorption of nutrients. So they are loaded with microvilli in the small intestine, and also sometimes the nephrons of the kidney that you'll see in the urinary system. And the other type of cell structure is cilia. So cilia basically move materials across the cell surface. So they beat back and forth to push material around. So they're present in the respiratory tract. And due to the presence of cilia in the respiratory tract, your lower respiratory tract, usually upper respiratory, cilia is present in the upper respiratory tract, and due to the presence of cilia in the upper respiratory tract, the lower respiratory tract does not get infected because these hair-like structures known as cilia trap all the dust and debris and prevent the lungs and the lower respiratory tract from infection. That's why when you wake up in the morning, you sometimes feel like you have to cough out some sputum or some cough. It's just because cilia has been doing a good job of trapping the dust and debris every time you breathe in something. So the next cell extension is the flagella or flagellum if it's single. So the only flagellated cell in the human body is the sperm. So the sperm needs flagella for movement, and these move in whip-like motions back and forth. And bacteria actually have a lot of flagella, but the only cell in the human body that contains the flagellum is the sperm, because they need to move in order to fuse with the female ovum. So these are the main cell extensions in animal cells that you need to remember.